Good morning children, welcome for today's biology class. So today we are going to study the chapter continuation biotechnology principles and processes. So mainly we are going to cover the topic tools of recombinant DNA technology. So here the tools of recombinant DNA technology are enzymes. The enzymes are restriction enzyme, ligase, polymerase enzyme, then vehicle or vector DNA, then passenger DNA which acts as host organism. These are the three tools of recombinant DNA technology. So first let us study regarding restriction enzyme. So this restriction enzyme belongs to a class of enzymes called nucleases. So these are of two types. One is exonuclease, other is endonuclease. Exonuclease, it cut the nucleotides at the terminal ends of DNA molecule, either 5' or 3' terminal end. Endonucleases, they cut at any point except terminal ends. Okay, so these two are restriction enzymes, restriction exonuclease or restriction endonuclease. Mostly used is restriction endonuclease. Stewart and Werner, these two scientists in 1963 isolated two enzymes from E. coli, so that were responsible for restricting the growth of bacteriophage. Among this, one of them added methyl groups to DNA, that you call that as modification enzyme, and the another one cut the DNA into segments called restriction endonuclease. Then other three scientists, Smith, Because and Kelly, so these three they isolated and named the first restriction endonuclease from Haemophilus influenzae bacterium, that was called as HIND2. So it always cut the DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs called recognition sequence. This sequence you call that as palindrome. So these sequences when you read from right to left or left to right, so the orientation will be same. For example, GAA TTC, it is from 5' to 3' end. If you read from 3' to 5', it will be CTT GAA. GAA TTC, and here 3' to 5' the same thing in opposite direction, CTT AAG. Okay, this is the thing we call that as palindromic sequences. So here different steps in the formation of recombinant DNA technology by the action of restriction endonuclease enzyme EcoR1, it has been given here. So here this is the vector DNA, it is the foreign DNA. First cut is made in both the places, then after that it has been joined by ligase enzyme. So then the recombinant DNA is formed. While making cut, two types of cut are there. One is sticky cut and other is blunt cut. In sticky cut, you see the cut will be made like that. So the enzyme acts in two different places. In blunt cut, in a similar place, and the cut will be blunt like this. So here it will be cross. It is sticky cut. So actually we prefer sticky cut comparing to blunt cut. Okay, so here after making the sticky cut then it can be made joined. So how is it joined? By using the DNA ligase enzyme. Okay, so first the cut is made in the two strands at specific points. Then later after making the cut, so then it is joined by the enzyme DNA ligase. Okay, this is the thing, the entire what we studied previously also, the steps in DNA recombinant technology. First we are selecting the desirable gene, and the desirable gene is inserted inside by making a cut, and it is joined by using the ligase. It is introduced inside the E. coli, the plasmid, then it is made to divide. Then ultimately whatever we need, the modified organism we get. So how do we give name to the restriction enzyme, nomenclature of restriction enzyme? The name of enzyme it is derived mainly, step by step let us see. Mainly we are naming from the organism from which the enzyme is isolated. the first letter of the genus becomes the first letter of the enzyme. Okay, it is written in capital letter. Then the first two letters of the species make the second and third letter of the name of the enzyme. So they are written in small letters. So these letters are written in italics. Then the fourth letter of the enzyme is the first letter of the strain. So it is also written in capital. Then the Roman number written at the end of the enzyme indicates the order in which the enzyme was isolated. So let us take example ECO R1. It is extracted, isolated from Escherichia coli. So that's why we name in this way. The capital letter E from Escherichia, then CO from coli, then R from the strain RY13, then Roman letter number 1, it indicates the first enzyme isolated from the bacterium. Okay, so how the enzymes are named, give example like that. No, it can be asked in 2 marks or 3 mark questions. So you learn this, children. And even palindromic sequences, what we studied also, no, that can also be asked. And the diagrams, both the diagrams, whatever we studied, these diagrams also can be asked separately in exams. So you practice this diagram also. So now let us see the separation and isolation of DNA fragments. The cutting of DNA was done by restriction endonuclease. Okay, then these fragments can be separated by a technique called gel electrophoresis. This also can be asked separately, how the separation and isolation of DNA fragments can be done. Otherwise, the technique of gel electrophoresis, like that they can ask. So the DNA fragments are negatively charged molecules. So that's why they can be separated by moving towards anode. Okay, so we are using the medium agarose, that is natural polymer extracted from seaweeds. The DNA fragments, they separate according to the size while separation. The smallest fragments, they will move further, faster, far away, and the largest fragment will be staying closer to the well. So here we are using the staining substance called ethidium bromide. So on exposure to UV radiations, it will provide orange colored band. So this method you call that as elution. So let us see the diagram, you will understand. So here these are the wells, 1, 2, 3, 4 wells are there. So here we are feeding the DNA fragments, whatever isolated. So when this is the agarose medium, so when we are passing electric current, then automatically the movement of the DNA fragments occur. So while moving, the larger fragments will be closer to the well, and the smaller fragments, they move far away as they are light in weight. Okay, so and later it can be identified by ethidium bromide. So this method you call that as elution. Okay, children, so here you can see in orange colored band, how is it visible? The ethidium, as we are we stained by ethidium bromide, it is visible under UV light like this. Next, let us see regarding cloning vectors. So plasmids and bacteriophages are commonly used vectors. Okay, so already we studied regarding ORI, that is origin of replication. So this is a sequence of base pair on DNA where the replication starts. That's why you call that as origin of replication. So here when it is linked, any type of DNA, when it is linked to this sequence, then it will multiply. Okay. Then we are using some marker genes also. That marker, if we are attaching marker along with that, then automatically we can identify the presence of the gene, whether the gene what we attached is present or not. So here we are using the marker, selectable marker. By using the marker, we can identify the transformants from non-transformants, where we have introduced the new gene or not. Easily we can identify. When we introduce the new gene, The desired gene, we call them as transformants. In non-transformants, the transformation not occurred. Okay, so that the new gene won't be there. So easily by using the marker, we can identify. How you are highlighting by means of marker while you are studying, then easily you can identify the points the same way. So here we are using some marker. So what type of marker can be used? So here we are using the antibiotic resistant gene as marker. Okay, so here mainly antibiotic resistance, we are using ampicillin, chloramphenicol, tetracycline, like that. No, some antibiotic resistance can be used as marker. So when using this marker, easily we are attaching this gene, antibiotic resistant gene, along with the desired gene. Then easily we can identify the transformants from non-transformants, and only we can segregate the transformants. So by this method, using the selectable marker. Okay children, so thank you. We will meet in the next class. So till then, whatever we studied, gel electrophoresis and the tools needed for biotechnology and all, you study thoroughly. We will meet in the next class.